Okay, I wanted to do a really quick video before you did your Teosinte activity. And I want to talk about the ratios that I showed you and asked you to memorize. And if you remember, we had 3 to 1 and 1 to 1. These are for monohybrid crosses. And if your ratio is 3 to 1, that means that both parents are heterozygous. If your ratio is 1 to 1, that means one parent's heterozygous and the other parent's homozygous recessive. These are phenotype ratios. And what it means if this works out okay. In other words, if you do a chi-square analysis and you accept the null hypothesis that a 3 to 1 ratio in your offspring means that these are the parents, your null hypothesis is basically the outcome that you see in a Punnett square. That means that on a single trait problem, that means that these traits are controlled by a single gene. In other words, let's take sickle cell anemia, for example. Sickle cell anemia is due to a point mutation, and it's an autosomal recessive trait, meaning that it's recessive in nature, and it's found on a chromosome that's not the X or the Y. And when you get the ratio that you expect when you're looking at sickle cell, it's because sickle cell anemia is controlled by one gene. Most traits are not controlled by one gene. The vast majority of traits are controlled by multiple genes. That's what the Teosinte activity is going to show you. And then another thing that I wanted to go over was the other ratios. So the 9 to 3 to 3 to 1 ratio means that these are for two trait problems or dihybrid. A 9 to 3 to 3 to 1 ratio means that both parents are heterozygous for the two traits that you're looking at. And a 1 to 1 to 1 to 1 ratio means one parent is heterozygous for both traits, the other parent is homozygous recessive. This is called a test cross or a back cross. And what that means is that you're taking an individual with traits that you're looking at, and you're seeing the dominant trait, but you don't know what the second allele is. By doing a test cross, you'll be able to determine what that second allele is because of the ratios that you get. So that's why that's called a test cross. It's used to determine the makeup of a heterozygous or homozygous dominant group. So anyway, what these two ratios mean is one, independent assortment is occurring. And the reason that independent assortment occurs is because these two traits, trait A and trait B, or the genes that they are associated with, are on different chromosomes. And what you're going to find is if the ratio doesn't match. So for example, if we say that the parents are this, heterozygous for two traits, homozygous recessive, we're looking for a 1 to 1 to 1 to 1 ratio. But what if our offspring look like this? For the dominant, we have 540 individuals. For the recessive, we have 492 individuals. For an individual who has one of the dominant traits and the other recessive, so maybe they have the dominant A trait and the recessive B trait, 16. Or maybe they have the recessive A trait and the dominant B trait. So what's going on there? That's clearly not a 1-to-1 to 1-to-1 ratio. It's also obviously not 9-to-3 to 3-to-1. So if we're saying that these are the parents and we're expecting this ratio and we don't get this ratio, what this means is that independent assortment is not occurring, and that's because the two genes are on the same chromosome. We are saying that genes, the gene for A and B, are on the same chromosome. Another way to say that is that they are linked. That'll be an important concept when we start moving on after spring break. Another thing that I want you to notice about this, your big numbers where most of your offspring are, most of your offspring look like each parent. So most of the offspring either have both dominant traits like this parent or both recessive traits like this parent. These are the recombinants here. And the reason that they exist is due to crossing over, which is kind of talked about in your POGIL. So we'll go over this a little bit more after spring break, but I wanted to give you some of this information up front before you start working on the Teosinte activity.